My name is Salman Khetani. I am an assistant professor at Colorado State University. Before that, I was the co-founder and director of research at Hypergen Corporation, which is a small company in Boston that specializes in creating engineered liver models for drug testing. Basically, the current toxicology in vitro in human relevant systems, and again, we want to use human relevant systems because animals are not able to completely predict the liver responses. So if you use human relevant systems, current human liver systems, one gets less than 50% sensitivity with respect to detection of toxic compounds. So you might as well just flip a coin. Why even do the test? We're trying to improve upon that, and we've already improved it to 70-80%. The idea that if you ran a compound, there's a 70 to 80% probability that you will correctly identify it as a toxin so that it doesn't make it to patients and harms them as well as the marketplace. And so it's a more sensitive assay. Of course, it's only 70%, so we're continuing to do research to try to bring it as close to 100% as we can. When I was running Hypergen Corporation, we had a company come to us where they had 2 drugs in the clinic for hepatitis C, and they caused, in combination, pretty severe enzyme elevations in patients. But all of their animal testing had not shown anything. And so it was happening in patients over several weeks. And so they came to us with a human relevant system that could survive that long. So you could actually do chronic dosing of drugs. And we helped them figure out that actually one of the 2 drugs was problematic and the other was fine. And so that data was submitted to the FDA, and the FDA allowed reinitiation of the trial for at least one of the drugs. Those patients could have died if they had continued on that multi-drug regimen. And so having these in vitro assays may jumpstart a drug discovery program that previously was dead. I work on a model called IMPCC. It's basically micropattern co-culture where you have the hepatocytes, in this case the I-cell hepatocytes, that are patterned in very specific domains. And so this model system, what we're trying to do is use the CDI cells so that we can have a robust, nearly inexhaustible source of hepatocytes that one could use in this model system to try to screen for drugs much earlier on in the drug development pipeline so that patients ultimately don't come in harm's way. And with our model systems, we are finding that whether it's with the I-cell hepatocytes or hepatocytes from patients, we can actually get much higher sensitivity for drug toxicity detection than afforded in more traditional model systems. In my lab, we're actually working with primary hepatocytes, hepatic cell lines from cancers, as well as I-cell hepatocytes from CDI. And what we're finding is that the I-cell hepatocytes are actually much better in their liver functions than the cancerous cell lines in our model systems or other model systems. That's one conclusion we have come up with. The second is that the supply of hepatocytes from patients is so limited that ultimately we want to use a more reproducible, inexhaustible source. And so it's not so much a matter of which cell type is better, but in which particular application one is going to use it for. And the I-cell hepatocytes, I believe, will allow us to screen millions, thousands, if not millions of drugs in the future, whereas primary liver cells, hepatocytes, will be more useful for very targeted questions later on in drug development. We have designed the IMPCC model to be fully compatible with the way people do toxicology testing today, which means that today people use a standard 96 well plate or a 384 well plate or a higher format. They basically come in and they put drug in each well, and at some later time point, they assess what the cells are doing. From that workflow perspective, we have designed the IMPCC model to be exactly identical, meaning they get a plate that looks exactly like they're used to. However, instead of having cells randomly distributed at the bottom of each well, what you see are little dots of the I-cell hepatocytes or hepatocytes in general. You see these little dots surrounded by the fibroblasts, but from a workflow perspective, it's exactly the same as what they're used to. Before the iPSC technology, the liver field, as well as just in general the drug testing field, had either cell lines, the cancerous cell lines, or primary cells. And primary cells are not easy to obtain. So there's a big gap between primary cells and cell lines. So I feel like the IPS technology has filled that gap where you get a cell type that you can expand with nearly inexhaustible cell numbers, but you can differentiate and manipulate them such that they're better than the cancer-derived cell lines and closer to the primary cells so that you can test more drugs on it. Cellular Dynamics has been a very robust partner, not only in terms of provision of the the IPS technology, but just overall their scientific acumen and really working as a collaborator rather than just as a vendor, which is something that I really wanted to enable, because ultimately Cellular Dynamics knows their cells and I know what I do. So having that scientific collaboration is really the way to move the field forward.